Hepatitis is a term used to describe a condition in which there is inflammation of the liver. Hepatitis can arise from several causes, including chronic alcoholism and autoimmune diseases. However, the most common types of hepatitis are a result of viral infection. There are 5 main types of viral hepatitis. These 5 types share some commonalities but differ significantly in several aspects, including viral genome, route of transmission, incubation period, and serology profile. These 5 types are named A through E and arise due to infection by viral particles of different families. Each virus type has a different genomic structure which dictates individual variation in replication and life cycles. The mode of transmission also differs between the types, meaning that some, like B and C, are more common than others, and others, like type A and E, are more prevalent in developing countries where there is a greater risk of ingesting contaminated food and water. Viral incubation periods are dictated by the rate at which the virus enters the host cell and begins replication processes, as well as the viral load and the host susceptibility to the virus. There are also clear differences in serological markers that can be detected and used as diagnostic criteria to confirm viral infection. Of these 5 types, hepatitis B is one of the most common. Hepatitis B is a DNA virus of the Hepatinivirudae family. It can be transmitted through contact with infected blood, through use or exposure to contaminated drug use equipment, medical or tattooing supplies, and needle stick injuries. It can also be transmitted via exchange of sexual fluids through unprotected sex with a carrier, and also via vertical transmission, whereby the mother can pass on the virus to the neonate during the 3rd trimester of pregnancy. Depending on the route of transmission and the age at which an individual is infected, as well as variations in immune clearance capacity, hepatitis B can result in either a short-term acute infection or persistent chronic infection. It is estimated that there are currently more than 250 million people living with chronic hepatitis B worldwide, including approximately 230,000 individuals in Australia, which makes up 0.9% of the population. The prevalence of hepatitis B in Australia has a strong correlation with endemic countries of origin. A significant proportion of affected individuals are of Asia-Pacific origin, as well as Africa, the Middle East, and Europe. The other predominant groups affected are Aboriginal and Torres Strait Islander people, intravenous drug users, and men who have sex with men. Hepatitis B infection is an important cause of morbidity and mortality in Aboriginal and Torres Strait Islander individuals, who have rates of chronic infection at approximately 3.7% compared with 0.9% of the general population. These higher rates of chronic hepatitis B infection also mean that Aboriginal and Torres Strait Islander individuals are at a greater risk of developing long-term complications. The pathogenesis of hepatitis B is a complex process and is not completely understood, but is generally thought to involve an interplay between the virus itself and the innate and adaptive immune responses of the host. When the hepatitis B virus is contracted, the host's innate immune system is largely unaware of the presence of the virus. This is because the virus can act as a stealth virus by evading the innate immune response by not inducing any cellular gene expression as it infects the liver. This allows the virus to begin the process of replication undetected during the early stages of infection. The hepatitis B virus docks to a receptor complex on the hepatocyte cell membrane via envelope docking proteins, then enters the cell via receptor-mediated endocytosis. As it enters the cell, the core particle is uncoated and released into the cytoplasm. This core particle, containing relaxed circular DNA, is translocated to the nucleus, where it is converted into covalently closed circular DNA. This CCC DNA is then transcribed by RNA polymerase II and converted to pregenomic RNA. The pregenomic RNA is encapsulated before being converted back to rcDNA via reverse transcriptase. These newly transcribed core particles are then packaged into vesicles in the endoplasmic reticulum before being exocytosed out of the cell. As replication continues, the virus can pass into circulating blood and spread to other hepatocytes, increasing the viral load. The virus engages with different immune components as it progresses. However, the inactivation of the innate immune system means that there is a failure to induce early CD4 T cell priming, which is thought to result in functionally impaired CD8 T cell responses, which consequently leads to persistent infection. There is also evidence to suggest reduced activation of natural killer cells, interferons, and TNF-alpha, which inhibits cytolytic activity that could aid in the clearance of the viral infection. Thus, a dominant cause of the viral persistence of chronic HPV is the weak antiviral adaptive immune response. There are also additional factors that have been proposed that could be contributing to viral persistence, including viral inhibition of antigen processes and modulation of the responses to cytotoxic mediators. The clinical presentation of hepatitis B is not always obvious and varies between individuals. However, there are specific signs and symptoms that are associated with both the acute and chronic phases of hepatitis B infection. Acute infection often presents with feelings of nausea and episodes of vomiting, as well as loss of appetite and fatigue. These symptoms are generally attributed to inflammatory processes occurring in the body and an interruption to normal metabolic processes in the liver. As the infection progresses, individuals often experience pain and tenderness in the right upper quadrant of the abdomen due to an enlargement and inflammation of the liver. Individuals also tend to exhibit signs of jaundice and darkening of the urine. This occurs because the damaged liver is unable to effectively remove bilirubin from the blood as it normally would. Bilirubin is a yellow pigmented breakdown product of erythrocytes, and when its concentration exceeds normal levels, there is deposition into the tissues and the urine, resulting in jaundice and darker coloured urinary excretions. Prolonged infection with hepatitis B can also lead to further complications, including the cirrhosis of the liver and hepatocellular carcinoma, which are irreversible and potentially fatal.